Okay? Okay, so we're going to be taking a tour through the cell. I think a logical place to start is to figure out what are the minimum requirements that you need to be called a cell or to be considered a cell. What do all cells have in common? Pardon? Okay, you've got to have some kind of genetic information. Okay, and that genetic information is in the form of DNA and in its intermediary RNA. Okay?
Some type of structure, the membrane or the cell wall.
Yeah, there's got to be some kind of boundary structure, the cell membrane, that separates the environment from the inside of the cell. And that cell membrane has to be able to regulate what enters and leaves the cell. You have to have a cell wall. No. Cell wall is optional. Some cells have them, some don't. And don't confuse cell walls and cell membranes. They are different things. What else do you need? If I've got some DNA in a membrane, I get a cell, right?
Something that produces energy.
Okay, yeah, you need some way to produce energy. Okay, mitochondria do produce energy. Do you have to have a mitochondria, though, to be a cell? It turns out, no, you don't. But you have to have some energy-producing process. That's going to occur within the fluid of the cell. What do we call that fluid where all the biological molecules are kind of present? Cytoplasm. So you got to have some cytoplasm, okay? And then what's something else you need? You need ribosomes because you have to be able to produce what? What is the genetic information really information for? How to make proteins. And where are those proteins made? They're made on the ribosomes, okay? We need cytoplasm, we need DNA or genetic information, we need ribosomes, and we need cell membranes. We introduced already this concept of prokaryotic and eukaryotic cells, and I think it's important to be able to distinguish between those two fundamental cell types. Remember that if you are adopting the three-domain system of classification, which is the more recent system of classification, then there are actually two domains that are prokaryotic in nature. There's the domain prokarya, which are the kind of true or common bacteria, and then there are these weird archaea bacteria, these ancient bacteria. The halophilic bacteria falls in that particular category. And then the third domain are the eukarya, all those that are made up of one or more eukaryotic cells. Here are some fundamental differences between prokaryotic and eukaryotic. Prokaryotes tend to be smaller in size, simpler. Very importantly, they don't have membrane-bound organelles. And then these are examples, bacteria and archaea. Eukaryotic cells being larger, more complex because of the compartmentalization of their cytoplasm into these little working units that we call organelles. And those are the protists, the plants, the fungi, and the animals. Those four kingdoms in the four-kingdom system of classification. If you look at a prokaryotic cell under a standard light microscope like the ones we have in lab, they're very unimpressive. They're not nearly as cool looking as a eukaryotic cell. And that's led a lot of people to believe that these are just really kind of uninteresting, pretty boring, pretty primitive type of cells. Well, if you start digging into those cells in a little bit more detail, they really do have some complexity that you really can't appreciate until you start looking at them with something like an electron microscope. But there are cellular structures that are associated with prokaryotic cells, but they're not considered to be organelles. They're not these membrane-bound structures within the cytoplasm of the cell. So they can have a cell wall. Most bacteria do have a cell wall, but that cell wall is fundamentally different in structure than a plant cell wall. They can have flagella. They can have a structure called a capsule. They definitely have to have genetic information, DNA, but it's not enclosed in a membrane-bound sac. It's not a nucleus, okay? It's an area called the nucleoid. They might have this capsule. They have to have ribosomes. So these particular cells are pretty simple structurally. But where they put eukaryotic cells to shame is in terms of their metabolism, they are incredibly complex. Much more so than eukaryotic cells. Eukaryotic cells are a little bit more specialized. And as part of that specialization, they've kind of given up some of their metabolic adaptability. You worked with some bacteria last semester when you did the mouthwash experiment, right? And before lab, we transferred some bacteria into one of those tubes of broth, and they grew just very happily in that broth. I mean, a few cells gave rise to millions of cells overnight. That broth had very, very simple nutritional requirements. Could you live by just drinking that broth? No. You wouldn't get the right mixture of nutrients. Your cells have lost the ability to produce some of the molecules in their metabolism that bacterial cells can produce very easily. So simple structurally, but very sophisticated metabolically. One of the things that I would like to talk about a little bit is the fact that prokaryotic cells being small has some relationship to their metabolic rate. These particular bacteria can divide over and over again at a much faster rate than eukaryotic cells can. Something that we use to express how fast a cell can divide is a concept called generation time. With eukaryotic cells, it's not uncommon for a typical cell to have a generation time of a day or more. Some cells have extremely long generation time. Anybody know what the typical generation time of a bacterial cell is? Ballpark average is around 20 minutes. Okay, so they have a much faster ability to divide and replicate. And part of that is the result of the fact that they have a very high metabolic rate. Well, how's that related to size? Okay. Anybody know? Why something that is very, very small might potentially be able to have a higher metabolic rate than a cell that's big? Pardon me? Won't need as much energy. Okay, that's possible. Any other thoughts? Let's ask this question another way. Is there an upper limit to cell size? We know that there are lower limits to cell size. A cell can't be smaller than a DNA molecule, for example, because cells have to have DNA. But is there a limit as to how big a cell can be? Yeah, actually there is. And it relates to a concept that's really important. It relates to the concept of surface area to volume ratio. When we're talking about a cell, what cell part constitutes its surface area? Which of those things you have to be a cell would we equate with surface area? The membrane, right? Okay. What would relate to a cell's volume? Which of those cell parts? Cytoplasm. Okay. So think of it this way. There's all this metabolic activity going on in the cytoplasm. There are enzymes. There are processes like cellular respiration. Okay? And they require nutrients to support that metabolism. And then also there are waste products that are generated as a result of that metabolism. Where do the nutrients come from to support metabolism? The environment, right? Where do the waste products have to go? Out, back into the environment again. So what do they have to cross? The cell membrane. And we'll talk later on about this, but the cell membrane is differentially permeable. It lets some things cross, not other things. But the traffic of materials related to metabolism involves moving across the membrane. So if I've got a big old cytoplasm, I'm going to need a lot of nutrients to support that, right? That metabolism. So I'm going to need a lot of membrane surface area for the transport of those nutrients to move across. Okay? So there's a relationship between cytoplasm and membrane that's really critical and really important. And if that gets too out of whack, then you've got a problem for the cell. Well, if a cell increases in size, those two parameters really start changing at different rates. If I have a 5 times increase in size, the size increases as a product of the square of the surface area, increases as the product of the square of the size increase. Well, what does the volume increase as? As the square of the size increase? Volume is 3 dimensions, right? It increases as the cube of the size increase. So as cells get bigger, the surface area is getting bigger by the square of the size increase, but the volume's increasing by the cube of the size increase. So what happens to surface area to volume ratio? It gets smaller and smaller and smaller. And at some point in time, if the cell gets too big, then there's not enough surface area or membrane to support the metabolic activities occurring in the volume or the cytoplasm. So there really is an upper limit to cell size imposed by that surface area to volume ratio. Now, one of the strategies that organisms can use is to divide things up. This has a lot more surface area than this does. Okay? So one of the adaptations you'll see oftentimes in cells is folding of membranes, for example, to increase the surface area. Next week in lab, you will look at epithelial intestinal cells, right? Mm-hmm. And one of the things that you'll see is that those epithelial cells have all these little foldings in their cell membranes to increase surface area for absorption of nutrients. If that membrane were just smooth and didn't have those indentations, it would have far less surface area and far less absorptive abilities. So this idea of increasing surface area by folding or subdividing is a really pretty important concept in biology and cell structure. Okay, so here's an overview of the basic animal cell structures that we're going to look at. We're just going to kind of go through this list. And then we'll talk about plant cells, and most of these are found in plant cells as well. But additionally, we'll have to talk about the cell wall, the chloroplast, might mention the peroxisome as well. One of the tasks that you have is to basically do some word association and to know, what's the major function of this? And how is its structure as a cell organelle related to that particular function? Same for ribosomes, same for this. Okay? So let's start kind of bopping through our tour of the cell. So here's the picture you've seen before, the static picture of the cell showing typical organelles that you might find. If we start from the out and work our way in, all cells have to have a membrane. We're going to defer our discussion of the cell membrane until after we do the tour of the cell. So we're going to talk about all the cell organelles first, and then we're going to come back and in Chapter 5 talk about membrane structure and function. The structure of a membrane is incredibly related to its function. And the major function of the membrane is to what? To regulate what enters and leaves the cell. So here's where you'll see some application of some of the biological molecule stuff that you're reviewing right now. For example, the cell membrane is a phospholipid bilayer, and the nature of those phospholipids is very important in determining what can enter and leave the cell and why that membrane is differentially permeable. Organelle membranes also consist of phospholipid bilayers. So we're going to come back and talk about that particular cell structure in a lot of detail later on. And it's what, the third lab, I believe, that we do the membrane structure and function. Okay. So I decided not to pull out clickers today for this. What's contained in the nucleus of a cell? If you want to respond, you can use your fingers as a response. 1 through 5 for fingers. So which of these are found in the nucleus of a cell? They all are. Okay? So what should our concept of the nucleus be? If you could try to explain to somebody, what does the nucleus be? What would you tell them? Okay, it's the storehouse of genetic information within a cell. Sometimes the concept is put forth that it's kind of the brain of the cell. I don't really particularly like that analogy because it evokes all these ideas of the nucleus being able to think and stuff. What were you going to say?
Obviously, it has the instructions that are for everything the cell does, every function.
Yep. It contains the instructions for all of the things that make up a cell. One of the concepts that we're going to talk about in much detail later on in the course is the concept of the central dogma. Did you guys talk about central dogma last semester?
Very basic.
Very basic. Okay. Does anybody know what a dogma is?
Like a general idea that's considered like concrete.
Okay, yeah, it's a belief. Okay. So this is the central belief of biology. And what does the central dogma say, very simply?
DNA transcribes RNA that transcribes proteins.
DNA is transcribed in RNA, and RNA is translated into protein. Okay? So which of these are associated with the nucleus? Well, the DNA is found within the nucleus, right? Does the process of transcription take place within the nucleus? Yeah, it does. It does. DNA is turned into RNA molecules. What are some examples of types of RNA? mRNA, rRNA, ribosomal RNA, and tRNA. And later on this semester, we're going to see that there are some other kinds of RNA too. There are those big three, but then there's some other ones we've discovered since then. So this happens in the nucleus. Where does translation occur? Does that happen in the nucleus? No. Where does it happen? It happens on the ribosomes. And where are the ribosomes located?
Endoplasmic reticulum.
Okay, they might be attached to the endoplasmic reticulum, but they also might be floating around in the cytoplasm, if you like. Okay? Free and membrane-bound ribosomes. Talk about those concepts? Last
Yes.
semester? Okay. So this is a cytoplasmic phenomenon. So there's going to have to be some traffic going on between the nucleus and the cytoplasm. RNA molecules are going to have to move out of the nucleus into the cytoplasm. As it turns out, there are some proteins that are going to have to move from the cytoplasm into the nucleus because ribosomes are actually manufactured in the nucleus. So there's this traffic going on. So one of the things you'll notice is that if you look at the nuclear membrane, there are these interesting little passageways called nuclear pores, which can allow molecules to move across that nuclear membrane. Another thing that you'll notice is that some of the cell organelles, like the endoplasmic reticulum, oftentimes are continuous with the membrane of the nucleus. You guys talked about mitosis and meiosis last semester, I know. What's one of the dramatic things that happens to the nucleus of a cell prior to prophase? Or even during prophase? Prior to prophase, what happens to the genetic information in the nucleus during interphase? The chromosomes replicate, right? During that S phase of the cell cycle, remember that? Pardon me?
Is it the genetic material condenses?
Genetic material starts to condense in prophase. What's another thing associated with prophase besides the genetic material condensing and the chromosomes getting... Pardon me? The nuclear membrane breaks up or disappears. Remember that? Kind of get more room to work and to sort out chromosomes. Well, where's that nuclear membrane disappear to? It doesn't just go poof. In reality, what happens is the membrane material that was originally part of the nuclear membrane is kind of reabsorbed temporarily into the endoplasmic reticulum membranes. And then during telophase, what happens? That nuclear membrane reappears, right? So that membrane material will then be moved back from the ER and reconstitute itself back into a nuclear membrane again. So there's some interesting dynamics that take place between membranes of various cell organelles, and they fuse with one another, they join one another, they split off one another. We'll talk about that more in a little bit, in just a second. There's a lot of interesting architecture within the nucleus of a cell, and there's some interesting interactions between chromosomes and things called scaffolding proteins that attach chromosomes to the membrane of the nucleus. But right now, your big association really should be the relationship of the nucleus to the central dogma and the fact that your genetic information will be transcribed into RNA, and then that RNA has got to get out of the nucleus and into the cytoplasm, and the importance of those nuclear pores in regulating that flow of traffic across the membrane. So, what's the function of ribosomes? Well, we've already talked about this. Okay. Ribosomes are involved in protein synthesis. I like to refer to them as the workbench of protein synthesis. Anybody have a workbench in your garage at home? Go out there and, put some good things together, hammer some things together. What's assembled on that workbench of the ribosome? A polypeptide chain is assembled. A chain of amino acids bonded together. What's the name of the bond that joins the amino acids together? Peptide bond, okay? What are the building blocks that the polypeptide chains are made out of? Amino acids. Okay? So the ribosomes are where the amino acids are hooked together. It's kind of a structural unit that facilitates grabbing the correct amino acids and joining them together to make the polypeptide chain. What determines the primary structure or the sequence of amino acids in a given polypeptide chain?
The R root.
They have different amino acids, have different R groups, but what specifies the actual sequence of the amino acids in that polypeptide chain? Where's that information encoded? It's encoded in an RNA molecule, but that RNA molecule got the information from a DNA molecule, and a particular section of that DNA molecule we called a gene, right? Okay. So the DNA information, the genes, specify the primary structure of a polypeptide chain by saying, if you want to make this protein, you put this amino acid together with this one, together with this one, together with this one. The actual assembly occurs on the ribosome. So, here is an electron micrograph showing you a segment of cytoplasm of a cell. And all these little black dots in here represent ribosomes. And some of them are bound to membranes like the endoplasmic reticulum. Others are floating free in the cytoplasm. If you look at a ribosome, this is the representation of ribosomal structure. And ribosomes consist of two pieces or subunits. A large one and a small one. But from a molecular standpoint, what is a subunit made of? What are the biological molecules that make up those large and small subunits? Are they DNA? Are they carbohydrate? Are they lipid? Are they protein? Ribosomes consist of two types of biological molecules. RNA, specifically ribosomal RNA, and protein. And those molecules, both of which are polymers, okay? A chain of amino acids folded up. A bunch of nucleotides joined together, folded up. And those molecules interact with one another to form these structures called the large and the small subunits. Now, that raises a very interesting question if you think about it. Because ribosomes are made up of ribosomal RNA and protein. But what do ribosomes produce? Protein. So where did the protein come from that's needed to make ribosomes if ribosomes are needed to make protein? It's an interesting question. Yeah. Okay, true or false? There's a difference in the makeup of cytoplasmic eukaryotic ribosomes and prokaryotic ribosomes. Show of hands, one or two, true or false? There's a difference in prokaryotic and cytoplasmic eukaryotic ribosomes. I see a couple brave fingers. I see a 1, I see a 2. I see a lot of people who don't know. Another 2. The answer to this question is true. Yes, there is a difference. And that difference is expressed in terms of something called an S value. If you're interested, don't have to know this. S stands for Svedberg, the person who discovered this idea. S values reflect differences in size and shape of molecules. So if you have a different S value, there's different size and shape to those molecules. It's actually determined by spinning molecules in a centrifuge and seeing how far and how quick they move down that centrifuge tube. Eukaryotic cytoplasmic ribosomes have an S value of 80. Prokaryotic ribosomes have an S value of 70. Now, both the ribosomes have large and small subunits. Okay, there's still those, two pieces. But there's going to be some difference in the proteins and the ribosomal RNAs that are in this ribosome than there are in this. And that's reflected in their different S values. Well, at this point in time, you might go, why are we talking about this? This is just minutia. What can be consequential about spending some time talking about this? Well, actually, as it turns out, there's a very important medical application to this difference in eukaryotic cytoplasmic and prokaryotic ribosomes. Anybody know what it is? Has anybody been sick recently? Has anybody taken antibiotics recently? Okay. Well, as it turns out, there are a couple of major classes of antibiotics. One class or class of antibiotics called cell wall inhibitors, and they interfere with the formation of a bacterial cell wall. And if you interfere with the formation of a bacterial cell wall, it can kill the bacteria. Do these antibiotics affect your cells? Do you have a bacterial cell wall? No. Okay. So that's pretty cool, because that means that these particular antibiotics can be pretty selective. They'll kill the bacteria but won't kill your cells. Does anybody know some examples of cell wall inhibiting antibiotics? Have you ever taken penicillin before? Okay. Penicillins and cephalosporins are two major big groups of cell wall inhibiting antibiotics. And there are all kinds of different penicillins. Probably when you were little, you might have taken amoxicillin if you had an ear infection. Probably as a pink liquid. Well, there's another strategy for selectively killing bacteria and leaving your cells unharmed. And that's to interfere with protein synthesis. Because if you knock out protein synthesis, then that kills the cells. But how can we selectively kill bacterial protein synthesis and leave our cells unharmed? By exploiting this difference in ribosomes in prokaryotic and eukaryotic cells. The protein synthesis inhibitors knock out protein synthesis on 70S ribosomes, but do not affect 80S ribosomal protein synthesis. So if anybody's ever had erythromycin or tetracycline, these are examples of a very long list of protein synthesis inhibiting antibiotics. Why did I specify here cytoplasmic ribosomes? What does that imply when I make that statement?
The ribosomes on the reticulum are maybe different size.
That's a possibility, that the endoplasmic reticulum membrane-bound ribosomes might be of a different type than the free ribosomes. What do you think? Is that true? Membrane-bound, free ribosomes different in S value? As it turns out, no, there's not. They're the same. But that statement implies that there are some different kind of ribosomes in the cell, that there's something besides cytoplasmic ribosomes. Does anybody know where else you find ribosomes in a eukaryotic cell besides the cytoplasm? Yeah.
It's actually endoplasmic reticulum.
Not an endoplasmic reticulum, but in another cell structure.
Aren't they on the nucleus as well?
Mitochondria. Mitochondria have 70S ribosomes. Mitochondria also have DNA. That is part of the evidence that is in support of the idea that mitochondria and chloroplasts, if you're a plant, actually arose from bacterial cells that invaded primitive eukaryotic cells and developed a partnership with those cells. So that your mitochondria and chloroplasts are actually have bacterial ancestors. That particular idea is a theory called endosymbiosis. So is there some potential harm to your mitochondria by taking tetracycline erythromycin? Potentially. Okay? Because it can inhibit mitochondrial protein synthesis. A normal 7-day prescription of antibiotics is not going to cause enough damage to your mitochondria to be of any consequence. But sometimes people are on very long-term antibiotic therapies. And if those are protein synthesis inhibiting antibiotics, you've got to be really careful. And the doctor's going to have to then monitor the situation and make sure that that inhibition of mitochondrial protein synthesis is not causing a problem. Okay, cells do secrete proteins. We have cells in our body that are part of important glands called endocrine glands. What do endocrine glands secrete? Hormones. And some of the hormones are proteins, like insulin. Are all hormones proteins? There's another class of hormones besides the peptide hormones. There are also steroid hormones, like the two that we showed on the picture when we were talking about functional groups. Estradiol and testosterone, they're steroid hormones. Proteins that are secreted by the cell are made on ribosomes. What kind of ribosomes? Are they made on membrane-bound ribosomes, or are they made on free ribosomes? One or two. Membrane-bound ribosomes. Okay? Free ribosomes produce proteins, but those proteins will stay in the cytoplasm of the cell where they're made. So, for example, in order for glycolysis to occur, you need enzymes. Those enzymes are needed right in the cell, in the cytoplasm. They're made on free ribosomes. If you're a beta islet cell of the pancreas, you need to secrete the protein insulin. You don't need it in your cytoplasm. It needs to be released out in the blood so it can bind to other cells. So those secreted insulin proteins are produced on these membrane-bound ribosomes. Initially, the proteins are made on ribosomes that are attached to the endoplasmic reticulum. What do we call ER that has ribosomes attached to it? Rough endoplasmic reticulum. What do we call ER that doesn't have ribosomes attached to it? Smooth endoplasmic reticulum. Smooth endoplasmic reticulum has some important functions. One of the things that it does is actually a lot of proteins that are made accumulate in the membranes of smooth endoplasmic reticulum, and some of those proteins are involved in functions like lipid synthesis, a number of the smooth ER proteins or enzymes are also involved in detoxification, trying to kind of clean up some of the metabolic wastes that are produced, for example, in the cytoplasm of the cell, or clean up some of the things that you ingest, like alcohol. What organ of the body is involved in a lot of detoxification of things that you take in? Liver. Okay. So what cell organelle would you expect to be very predominant in liver cells? Smooth endoplasmic reticulum. Okay. Well, the proteins that are made on the rough endoplasmic reticulum are eventually going to be transported out of the cell. But typically they go from one location to another during that transport process before they actually make their way out of the cell. Where's the first place that these secreted proteins typically go? From the ER to the Golgi, to lysosomes, to mitochondria, or to vesicles? Anybody know? In order to get to these two particular locations, or to actually attach to the cell membrane so you can be excreted, the first thing that typically happens to those proteins is that they're packaged up in little membrane-bound sac called vesicles. Think of the endoplasmic reticulum as one of those long balloons that you might see at a street fair. And inside of the balloon are the proteins that were produced by the membrane-bound ribosomes. At a street fair, sometimes the artist will then twist the balloon to make little animals. Well, let's say you were a really talented artist and you could twist that balloon enough so you could completely twist off a little smaller balloon that went floating away. That would be like a vesicle. And then that little balloon could go attach to another balloon in the cytoplasm, another organelle, and re-merge with it. And so whatever was inside the vesicle would now be in that new membrane-bound sac or balloon. Well, that might sound kind of impossible. And it would be with a regular old balloon, because balloons have static membranes. But cell organelles have fluid membranes. And because the membrane is fluid, if you pinch off a little sac, both will seal themselves off again. And when a little sac joins to another membrane, they'll fuse together because their membranes are fluid and join together and fuse together. So the next set of organelles that we're going to talk about make up a system in the cytoplasm called the endomembrane system. The endomembrane system consists of a series of cell membranes that are either physically connected or indirectly connected via vesicles. So the bottom line is that things can move throughout the cytoplasm by going from the membrane of one cell part to the membrane of another cell part because of that physical connection, or because a vesicle forms and joins one membrane to another temporarily. So what membranes are we talking about? Well, the nuclear membrane is considered to be part of the endomembrane system. So is the ER. And if we go way back to this one right here, see the physical connection that exists here between the ER and the nuclear membrane? So there's a direct physical connection. Vesicles can pinch off of the ER. Those vesicles can join with another membrane system called the Golgi. They can fuse together to form a membrane system called the lysosomes. They can fuse together to form a membrane sac called a vacuole. They can fuse with the cell membrane. And when they do that, whatever was inside the vesicle is released outside the cell. What do we call that phenomenon? Where things are moved to the membrane and then the membrane kind of pushes them out. That's called exocytosis. So, this is where we'll pick up tomorrow, as we'll look at the endomembrane system and talk about some of these other cell organelles, and particularly the flow of things between these various cell structures.